사람 처음 보는 거야? Antibody size is 150. 바꿔주세요. Okay, in immunocytochemistry, immuno means antibody, cyto means cell, chemistry means something. Okay, in the immunocytochemistry, you need to have antibody, maybe one antibody or two antibodies. If you need to use a two antibody, which antibody has a fluorescence? If you need to use the two antibodies, which antibody should have colorful material? It should be secondary antibody. If you have the fluorescence motif on primary antibody, then you don't need to use the secondary antibody. If primary antibody does not have any fluorescence motif, then secondary antibody can recognize the FDC motif of the antibody. FDC motif of the antibody, and then showing this fluorescence. Nothing with the T cell. This T cell, nothing with this immuno. This immuno means antibodies. You use antibody for visualizing your target. 이게 나온 것 같은데 왜 계속 답은 몇 개야? Who did the answer? I have 11 people here. Why I get only 10 answers? 안 되는 사람 누구야? 얘가 계속 처리한다. 오케이, GFP 뭐 공부한 거야? 공부한 거야? 안 한 거야? Size of GFP is 30 kilodalton. And it's a relatively small protein, but actually many proteins is even smaller than 50 kilodalton. So the advantage of the GFP is it can be used for live cell imaging, but there are actually a lot of problems. For some of you indicate that GFP looks like it's okay. It does not bother the original function of the protein. But actually, if protein is a 10 kilodalton, GFP is a 30 kilodalton. 30 kilodalton GFP, then expression of this protein can be affected by this GFP size, definitely. But I have seen tubulin was okay. Yeah, tubulin forms. Tubulin has an alpha and beta subunit. Each of them is a 50 kilodalton. And if GFP is added at this size, this should be adding into the microtubule formation like this. Right? And you need to be very carefully to determine where you attach this GFP. Either alpha tubulin or beta tubulin, usually alpha tubulin. And so you need to choose either C terminal or N terminal. In many cases, it cannot form a microtubule. So the scientists believe that approximately 70% of GFP fusion protein, they cannot fold properly. or they cannot locate into original position. GFP size is not trivial, it's huge. The 30 kilobyte size. How can we use the code part of the DNA? Well, you have a target protein, it's a gene, and you have a promoter, and your option is either to put GFP protein gene here or here, or even you remove those whole protein, just add the GFP gene here. Then, following those promoter information, the GFP is expressed in that of the cells. That's also possible. So this is a DNA editing. And Krylux systems can generate the rainbow, and it's not possible to have a neighbor neuron has the same color. Krylux is the one enzyme and the one of the location system. And then if you have CFP, BFP blue, CFP cyan, GFP green, yellow, YFP, RFP, and let's say 5 different genes. And then those can randomly cut and reconnect, and it can generate combinations of, if you have 5 genes and if you have random combinations, how many options do you have? Is it linear or job? Let's just say it's just linear computation. How many options do you have? Many, right? So then it can be many genes are randomly mixed up, then it can show green and red and cyan, and different combinations. By chance, neighboring gene maybe can have the same color, it's possible, but by chance, if there's 100, then out of 100 different colors, it can be, looks like randomly distributed. That's a rainbow. There's one more question. If this neuron divide and the other cell can express the same one or different color, well, once this combination is fixed, then that's done. If it's divided, it should have the same colors. So this recombination should happen in the middle of those division. Once it is divided, then recombination is also fixed. Then it should not make too many more cells. If they do, then you see the color mix. One big chunk, one color, and different. Okay. And there was some questions about calcium and pH, sensors, and GFP. This question was given by Hyunbin. So Hyunbin is going to present about this. He gave the question, and then he will give your presentation about this. Okay, welcome Hyunbin. Turn on my system. Say thank you to me.
Thanks. You're welcome.
So Hyunbin is going to talk about calcium sensor and then its application, the chemical calcium sensor and application in biology, and GFP, the version of calcium sensor.
Hello, my name is Hyunbin Cho, and today I'm going to present calcium indicator and GCAMP. In a bit more details, I want to talk about calcium sensors and signaling, how we actually measure the level of calcium in the cytoplasm of cells. So, if you got a cell in which calcium is going to rise, if you want to see a calcium signal, which is a calcium rise within the cytoplasm, and how are you actually going to see that? We know that calcium level outside the cell is much higher than the calcium level inside the cell. Calcium concentration extracellularly is approximately 1.5 millimolar, whereas calcium concentration intracellularly is about 100 nanomolar. You can use calcium moving from the extracellular to the intracellular component as a signal. When the calcium goes up, then it's going to change laws of downstream activity. Changes in the cytosolic calcium level can act as a cellular signal. It can change downstream signaling pathways because calcium is a second messenger we learned in class. A very handy tool for physiologists trying to experiment with calcium signaling is a way of measuring calcium level, a way of detecting when calcium goes up in the cytoplasm and where. So what we're going to do is create some sort of molecule that will be able to tell us when calcium goes up in the region of the cell, so that we are actually going to be able to see where calcium is going up and where you're getting calcium signaling. The main calcium sensor is a sensor called FURA2, and we can use it to measure the level of calcium in portion of the cytoplasm. It was developed in the 1980 by a scientist known as Roger Chen. So, how does FURA2 work? Initially, let's talk about it in the unbound state. FURA2, which is unbounded, it has no calcium bound on it. FURA2 is fluorophore. If it's sent in radiation of wavelength 340 nanometer, then FURA2 will emit radiation at a wavelength of 510 nanometer. So we're sending radiation which carries energy, and FURA2 will absorb a certain fraction of radiation. Now, there is another wavelength of light sending photons of wavelength 380 nanometer. Then FURA2 can also emit radiation again at this wavelength of 510 nanometer. Let's say we're saying the same intensity of radiation, 10 photons of radiation. 60% of photons get absorbed of 340 nanometer, and 70% of photons get absorbed of 380 nanometer. So if we send in the same intensity of radiation into FURA2 of these different wavelengths, we will get a different amount of radiation wavelength 510 nanometer coming back at us. Usually, if you plot wavelength along this x-axis, and if you plot the fluorescent emission on the y-axis, this one goes down and this one goes up. Remember this graph. So we've got an absolutely lot of FURA2 in the cytoplasm right now. And remember, it's not just about there being on an on-off state. So far, we've discussed that FURA2 can be in the two states, unbounded state and bounded state. When calcium is high, not all of them are going to have calcium bound to them. The level of calcium in this cell will determine what fraction of the FURA2 molecules have calcium bound and what fraction don't. A higher calcium concentration will increase the fraction of these FURA2 molecules that have calcium bound to them. The portion that are bound to calcium, when you stimulate this cell, let's say we stimulate with wavelength of 340 nanometer, the FURA2s that aren't bound to calcium are going to have a high fluorescence in response, because see in the previous graph, the ones which are bound to calcium in response to 340 nanometer will emit a low fluorescence. The actual fluorescence you will get back will be somewhere in between those two, because you've got a mixture. Furthermore, you could compare two fluorescence intensity by ratio like this. Take the fluorescence for 340 nanometer and divide it by the fluorescence for 380 nanometer. I believe that FURA2 emits more radiation at 340 than 380. From the ratio, you can deduce the amount of these FURA2 molecules that are bound to calcium. From that, you can deduce calcium concentration, the amount of calcium that's in the cell. So this ratio tells you information about the calcium concentration within the cytoplasm of the cell. It's because the ratio will depend on the occupancy of the FURA2 by calcium. How many of them, what fraction of them are bound to calcium, better than the absolute number. That's important because obviously your absolute fluorescence will depend on how many FURA2 molecules you have and how thick your cell membrane is. whereas the ratio between them won't depend on that. It's a nice measure for identifying calcium levels within cells, isn't it? We'll continue looking at our calcium sensors. The specific one we'll look at is a calcium sensor known as GCAMP. This is the ingenious little idea that Nakai had to improve signal to noise ratio compared to previous fluorescence calcium probes. Basically, what they make is a chimera. The chimera consists of green fluorescent protein, calmodulin, and calmodulin protein, M13, a peptide sequence from myosin light chain kinase. When calcium goes up, it is going to bind to this calmodulin. That looks like a convincing start. We have a calcium binder in our chimera. Then there are two halves of the green fluorescent protein, GFP, and we chop the green fluorescent protein in half and put the two halves next to each other, but sort of inverted basically. The next thing is GCAMP is the calmodulin binding domain of the myosin light chain kinase, MLCK. It is going to bind calcium calmodulin complexes, so the calcium calmodulin complex binding domain. Where do they get this binding domain from? They take it from a protein. We take the calcium calmodulin binding site of MLCK. So why does this work as a calcium sensor? This is all we need. When calcium goes up in the cytoplasm, calcium is going to bind to this calmodulin. When calcium binds to the calcium calmodulin complex, calcium calmodulin complex is going to want to bind to the calcium calmodulin complex binding domain from MLCK. So what ends up happening is that your chimeric protein folding like this, so that the calcium and the calmodulin complex binding to this calcium calmodulin binding domain, which we took from MLCK. And now the rest of the chimera is here. We have the two halves of green fluorescent protein, GFP. So at the moment, when it's in the resting state before, unbounded state, there is this GFP. It's split in two. Basically, and it's not functional. So if we shine UV onto this original protein, then we do not get green coming back at us. Then I have this whole protein. Basically, it's folded so that this calmodulin over here is now bound to the calcium. Firstly, to create a calcium calmodulin complex, and it is now bounded with the calcium calmodulin binding domain from MLCK. Now you have your two halves of this GFP very close together, and they now dimerize. Now end up with a dimer, basically a functional green fluorescent protein. So if I send UV of the right frequency, perfectly correct for this GFP, then what I'll get out is green light. So this is a way of visualizing calcium signals. When calcium goes up in the cytoplasm, and where calcium goes up in the cytoplasm. And when you shine UV on your cell, then where you're seeing green light coming back, that's where this protein has folded in this way and has made the functional green fluorescent protein. And where you don't see green light coming back, there's a low calcium and it hasn't folded. So this is how the GCAMP functions as a calcium sensor. We can visualize where calcium signals are occurring by where we are receiving green light back from the cytoplasm. Thank you.
If the calcium binding happens like this, GFP, RFP originates far away, and when calcium binding here is information changed like this one and this one. If it's close, then let's say energy transfer is more efficient. This energy transfer is called as a flat fluorescent energy transfer. So A and B case. Calcium bound form is more greenish or more reddish? Reddish. It should be more reddish because energy transfer happens more efficiently. This is gonna goes down, this is gonna goes up, they become like this. Okay. By looking at this ratio, then we can guess, we can calculate the calcium concentrations. So advantage of this GFP version is we can use this in live cells. But disadvantage is you need to express those gene inside the cell. Cell take up extra gene, usually the cell condition is not so good. So it's a very healthy cells. If you want to see, then you may need to use something else. Example of what can be showed was filled up two type of the dyes. You can drop those dyes. Then in the video you have seen, if you fire the neurons, then the neurons secrete calcium dramatically, and then you can see a bunch of different neurons. They are excited the neurons with high calcium. So that's kind of a current the measuring of which neurons are really activated. Thank you. Okay. 어쩌다 보니 벌써 마지막 날이네. So today, in addition to the video of the material I gave you, I prepared some of ongoing work, live cell imaging with the example of what I'm doing. So you can see some of those the current the work on this live cell imaging work. So our body is mainly our body is composed of different cells. Size of our cells, average size of our cells, 10 micrometer is kind of average. Bigger one maybe 20. The highest number is a red blood cell, occupying 70% of the population in our body. And maybe biggest cell is egg cell. Human egg much smaller than chicken egg, but still this is the 150 micrometer size. And whole length of the sperm is about the 50 micrometer. So head size is maybe smaller than regular cells. And maybe longest cell could be the neuron cells. And some of those cells, they have a different shape, but in many cases, the cells look like very similar to each other. Especially if you take out those cells from the body culture in the dish, and they look like just a round shape. If it's floating around like a ball, if it's attached to groin, then they look like this. So it's quite difficult to discriminate which one is which. So then how do we know whether those cells are different or not? And different cells, they have a different expression pattern of proteins, right? And maybe T cell, B cell, the lymphocytes, they look like similar by size and shape, but they may express the different proteins. Those specific proteins are proteomics. If you grind up the cell and take out the whole protein, then you may see in principle we should have thousands of proteins, maybe more than 10,000 of proteins. Total number of our gene, at least 25,000, and each different cells, they do not express all the protein, maybe half of them they may express. But amount-wise, some of them is very small, we don't see. So in this looks like a complicated picture, we have a highest number not higher than 1,000. Just some hundreds, still look like a quite complicated. Among them, we need to figure out which protein is expressed only in one type of cell. And if that is, then that protein we can call maybe biomarker of some protein, some cell. Looks like complicated, right? And this is a real example. This is T cell and B cell. Can you find out which protein is overexpressed in one type of cell? Not easy. If you have keen eyes, you can find something. This is both of them. Oh, here. Looks like this protein is only here. Not here, right? So this is a two-dimensional chromatography, and the chromatography data you got yesterday and today, it's difficult even if you overlap them, they may not exactly match, a little shifted, right? So then, even though overlap these two images, this still does not guarantee that we can get exact overlapping. One trick, after you get those protein sets, you can label those protein with one color of dye, all the proteins. The other protein with another color of dye, mix those samples and run them together. Then at least batch to batch variation you can minimize. Assuming that size of this dye is much smaller than protein, they do not interfere too much, then you can get this kind of images. This is green color, this is red color. If they are perfectly overlapping, then you may see yellow color. Right? But if you see red color, means these proteins are only expressed here, not here. Okay? If you see any green color, green, which is green, maybe this one may be stronger here than this. So using this kind of a trick, you can get help to figure out which protein is a little more overexpressed in one type of cells. Once you have that kind of the cell, that kind of a protein, specifically only one type of a cell, you call it biomarker. If you have a biomarker, then what can you do? Now, you know that this protein is a B-cell specific biomarker. One way is you can design or screen your probe which can bind to it, and maybe some of those probes they can change its colors or the intensity changes. Then those are sensors. Sensor is a small part of a special type of probes. Probes, if they bind to that the biomarker with a fluorescence, then at least we can report where they are. So in this case, the people say hypothesis-driven approach. I want to develop a probe for B-cell specific. Then you need to have a hypothesis. That hypothesis means most likely I know what is the biomarker of B cells. That biomarker gives you the hypothesis. Even though you have that biomarker, still design of those specific probe is not easy. So one very common biological way is this human protein, you inject it into mouse. Then mouse recognize this as antigen. Then they may generate a lot of antibodies. So those antibodies now collect them and then figuring out which antibodies bind to this most strongly, and you can get out single best binding antibody, so-called monoclonal antibody. Right? And utilizing hybridoma cells and then amplified, and then this is binding to this antigen biomarker, and this is antibody, and you label that antibody with the fluorescence. And here, this is a probe, and this is biomarker. In many cases, we have the idea or the concept that biomarker is huge, probe is small, but sometimes it can be opposite. Epitope for binding of antibody does not need to be huge, right? Even though the antigen is big, antibody can bind to different positions. Epitope is a very small part. Amino acid size was 3-4 amino acid size is enough size for antibody binding. So biomarker can be small and probe can be big. Once you have this kind of a biomarker, then you can apply them into the cell. And if that biomarker is on the cell surface, we call those kind of biomarkers as CD, cluster of designation or cluster of differentiation. It's some surface marker proteins. And so far, more than half a century of immunologists updating this the list over and over. Most recent one, about 10 years ago, they had a big meeting, and then, okay, we officially approved that 371 CDs. So we have learned the CD4, CD8, T helper cells, T killer cells, those are this biomarker name. CD4 cells, they have only CD4. Killer cells, they have only CD8. And those number of the CDs are up to 371. Sounds like a huge, but if you think about the total number of proteins inside the cell, they should be minimum thousands. Only those antibodies, the biomarkers developed so far is only 300 something. Partially because maybe those are surface proteins. If you count all three-dimensional space filling up of whole cells, maybe those biomarker number can go up a lot more. If you have antibody for surface CD antigen or the biomarker, you can recognize them. But if that biomarker is inside the cell. Problem here is the size of antibody 150K kilodalton, huge. It cannot penetrate this membrane. Membrane very thin, only 4 nanometer thickness. Antibody size was about the 10 nanometer size. Still, this antibody cannot penetrate this compact the membrane structure in lipostatus. To get in, you need to treat the cells, so-called fixing. Fixing, how do you do? Using chemical cross-linkers, like a formaldehyde or glutaraldehyde, 2-aldehyde structure, make cross-linking between proteins. Still, membrane is there, antibody still cannot get in. Now you add a detergent to dissolve some of those membrane. I said the detergent structure and membrane structure is almost the same. Liquid tail and charged head groups, detergent is the same structure. So by adding detergent, you can dissolve some part of the membrane. Now they have through those holes, now the antibody can get in and can recognize those target, whatever, biomarkers. Now you can define your target. And if your target is most likely those cell imaging, the nucleus you see as a blue color. Those are strong DEPI or Hurst-based, the DNA dyes are very well developed. So more than 90% of the cases, if you see cell images, blue color is designated for the nucleus. That represents that, okay, I'm a cell. Okay? And then you can define your target. In this case, this is true. Even though GFP can form up this kind of image too, but by adding antibody, we can show these tubular structures. This is a healthy muscle cells. Muscle cells that are connected together make this kind of tubes. Here, if you add a tubulin acting compound like the Taxol, then they attack those tubulin. You can see this breaking down. Our eyes, at least we have the RGB, 3-color receptor. We can recognize maybe 4 or 5 different colors. So actin red color, tubulin green color, this kind of image is possible. Cell is very thin, so it's optically transparent. But if you take out the brain looks like tofu, because of the high amount of the lipid molecule, you need to clear out to make it transparent. After that, if you add several antibodies, you can visualize those different cells. Rainbow is a biological way to show different neurons. In this case, just the 4 antibodies, they can show neuronal structure like this. Looks like it's really dynamic, and seems like we are observing the biological event, but actually to make that the images, you fix them by adding formaldehyde and then dehydrate. There should be snapshot image and also artificial images, dead images. So to overcome that, one possibility is using GFP. To do GFP, you need to do biological transfection, which takes a lot of work too. Other option can be maybe probes. For live cells, one thing you need to think about is the plasma, the membrane potential. I think we have talked about this before. Those are composition of phospholipid inside and outside, but they are not symmetric, they are different. And they say outside is positive, inside is negative, but none of those phospholipid heavy groups are positive. That means most likely inside is more negative than outside. So surface of a membrane, inside and outside, both of them are negatively charged. That means if you use negatively charged molecule, they cannot access to the surface. They will bounce off the surface. That's why negatively charged molecule, drugs, probes, usually the uptake is very poor. And if it is a positively charged, at least approaching to the cell surface, that's fine. They may stay here. Now, penetration of this thickness of 4 nanometer, size of this molecule may be a little less than 1 nanometer. So 1 nanometer molecule needed to cross that much of the lipid area. That depends on partition coefficient. Lipinski rule of bigger than 2 and less than 5, that's the range of reasonable hydrophobicity. If it's higher than this, they are stick to any membrane and they will not move. That's why if you inject any molecule here, if they have a strong pain and strong color here, that means too hydrophobic. They attach to all the blood vessels here. They do not move in. And if it's too hydrophilic, they cannot penetrate this at all. All of them will stay outside. So this reasonable or the range is important. And then if you can control that, then they can find its targets like mitochondria, Golgi, or DNA. That's something what you have seen for organelle selective probes. That part has been developed over last half a century. So reasonably, we have a good markers, good probes for nucleus binding, mitochondria binding, or Golgi binding kind of things. Now, that part is done, but if you think about how to discriminate the different cells, if you scale up your target, maybe the next level is maybe your organ. I want to see only liver. I want to see only heart or lung. So that kind of level that you need to think new hypothesis or the biomarker you need to have. So for that, maybe one possible way is you utilize antibody-like sets. Those kind of sets we call library. Collection of all those molecules we have seen the cDNA library. Right? So maybe those kind of a library, in terms of this kind of small molecule, fluorogenic or the chromogenic, two possibilities. One is inorganic molecule, transition method. The other one is elongated conjugation to observe the light. Inorganic way, transition state, transition methods, usually they are toxic. So usually we avoid to use those transition methods in our body. Then leftover option is this kind of the conjugated molecule which can absorb the light. But originally those molecules has a flexibility, then they can absorb the light, but they lose their energy by vibration or heat generation. Only when they bind to the biomarker, the motion can be suppressed, and this whole molecule becomes rigid, and they can become fluorescent. So example is like this. This is the highlighter of the molecule in your Hemang pen. The green color, maybe biggest consumption in human the society right now. The molecule structure is like this. Orange color Hemang pen is like this. Here, this is a fluorophore. Because of this carboxylate, they cannot rotate. This motion suppression makes this dye very bright fluorescence molecules. If you remove this small group, now they start to rotate. These small changes make the contaminant of this dye from 100% to 5% or 1%. So this dye is very thin molecule. Only when the biomarker binds this molecule and suppress this motion, then they can be turned on. So based on that idea, the small molecule fluorescence or fluorogenic library can be generated. And then if you keep those molecules in this kind of 96 plate, and first one and last one is empty, then 10 by 8, this is 80 compounds kind of a molecule you can construct. Here, since this is dim and this is brighter, but you don't need to think about this is maybe better than this. Because if it's bright, then only option it can be changed is going down. If it's dim like this, then once it binds into the biomarker, it can be turned on. Then low background and high the signal you can achieve too. So utilizing this kind of molecules, maybe thousands of them, and then you can apply them into the different cells. Let's say your background is chemistry or chemical engineering, maybe biology, but I don't know enough about the biomarker. These cells are just recently discovered. Then still, one possibility is utilizing this library, like antibody cells, and apply them into the cells. To make those the registration simpler, same format you can apply 96 well into 384 well format. Then one compound can be changed into 4. And one way is you can grow A cell here, B cell here. You can apply maybe twice, duplicate experiment. Then maybe you hope that, okay, A cell is not staying at all. Only B cell is staying. Then this probe can be B cell selective probe. So using this approach, still this cells are live state. In the live state, we have a chance to figure out B cell selective the probes. If that B cell is important cell, then this is maybe better or easier to use than antibody or GFP expressions. So those kind of examples, we have done maybe more than 30 examples, but I cannot show you too many. So today I will show you just 2 examples. We have learned about the tumor microenvironment. Once those tumors break up our first line defense, they make this chunk. Then the immune cells still they did not fully gave up. Still they try to control these tumors. But vigorously attacking those tumors can generate more problem. So it seems like they are negotiating with the tumors. So they just keep this boundary of the tumors. And seems like seemingly they are helping those tumors. If you don't cross this the police line, then we are going to protect you. And even they suppress those immune response around this area. So looking at this kind of situation, it's difficult to say whether they are still our policemen or they are spy to the tumors. This strange situation, which looks like a more favorable to tumors, this is what we call tumor microenvironment. Okay? So here, one example can be tumor initiating cell. Once we have a tumor, then the big chunk of those tumors, they don't have ability to make a new tumor in different sites. Even though we remove 99% of tumor chunk, still hidden very small amount of tumors, they revive and they regenerate the tumors. Those kind of tumors, we believe there are tumor stem cells or so-called tumor initiating cells. So if we wanted to cure the tumor completely, we needed to attack tumor initiating cells, so to remove tumor stem cells. So to do that, let's just say this is a regular tumor, just a big chunk, faster growing. And then this is a tumor initiating cell, and grow side by side. And then find out which compound, which is also bound to this. Tumor initiating yellow was found. And again, blue color is nucleus. Cells are there, but regular tumors, they are not staying. Only tumor initiating cells, they are staying in red. Looks like it's working okay. If you take out those tumor initiating cell, and then if you put that cell in new mice, even single cell, they can make a tumor. One cell can make a tumor. That means that's a really active, realistic tumor stem cells. They exist. And that means the probes does not affect the power of a tumor stem cell. It's a bad power, but still for research we need to keep them alive and aggressive to study. So finding out those is half of those work, and it's a useful tool. But as a research, if you wanted to publish this work, then the other half is you need to explain why this probe is specifically binding to which target. If you had an antibody, you already knew your hypothesis, right? You have your biomarker, that's why you took it and injected it into the mouse. You have your answer in hand from the beginning. But in this case, you put your tool first, and you don't know which target the probe binds. So most of those cases, the most common approach is so-called affinity matrix approach. Affinity means is the affinity between the probe and your target. It looks like it's fishing. Actually, that's really fishing. For those fishing experiments, your approach is you prepare this kind of bead, hydrophilic agarose bead or Sapadex bead, carbohydrate bead. Size of this bead is approximately 100 micrometer. It's much bigger than your cells. So that means, and then on the surface you coat your probe or drug molecules like this. And then you assume that in biological system, this is cytosol. In the cytosol, their proteins are swimming around. They should be healthy and intact, properly folded structure. And which fish bind to your bait is the role of the affinity matrix. But problem is the bead is much bigger than cell. Then you cannot put those beads inside the cell. That means you prepare your bead outside and you grind up your cells by adding detergent, break up the DD cell membrane, and collect those proteins and enrich them, and then apply them into this bead. Then through this process, by adding detergent, originally compartmentalized into different parts of the cell, they are mixed together, and they will not be happy. Half of those proteins may be denatured and exposed to their hydrophobic area, and then they hope that this binds to this bait, but maybe expose the belly, maybe it's just a touch. Non-specific binding happens a lot. So this technique is called affinity matrix, but this is the best method utilizing small molecule bait, like a drug molecule, to find out its target. So we thought we could do better because our compounds are not drug. These are the fluorescence molecule, and we already demonstrated the molecule goes into the live cell without the intact, without disturb those original function of tumor initiating cell, still alive and active, and then binds it to the target. So that means maybe this one molecule structure is this fluorescence part, and this part is about the chemically active motif, chloroacetyl group, which can bind to cysteine by forming covalent binding. Then using this probe, it's like this kind of a probe with an active warhead. Then we can send this probe into the seed of the cells. Then it goes into the cell, and then maybe if this is our target protein, then it goes into the cell and it makes covalent binding to the target protein. Once its covalent binding is formed, still this cell is intact, and protein was all compartmentalized into different area. And once it's done, then you can break up the cell and run chromatographic separation. Then you can see your target protein fluorescently. Then you can cut it out, and then mass spec analysis can tell you the sequence of this protein. Then you can figure out the binding target. So in this case, the binding target was denatine, which is already known biomarker for metastasis. So this was a happy case, but when we did this the work over and over, sometimes we can get this kind of target. But in many cases, you see, about 10% of bound protein, 90% of those probes are free molecules. Free molecule means those are filling up this cytosol without binding specific target. If this is 90% and the bound protein is only 10%, can you really say this 10% of binding is a real target of this? That's a questionable. And for a long time, people believe that this probe should have a binding target. Maybe that concept is wrong. If there is any transporter which can pump in, then accumulate those probes inside this cell, then may be enough. It doesn't need to bind to anything. Just it is a substrate of this transporter. Then if this transporter is responsible target, but different kind of target, not binding target. This is gating target. Then that possibility is there, we thought. And that example we proved using these cases. Macrophage is one of our immune cells, which is not the adaptive immune system. They bind to randomly to the bacteria or tumors. And they have, seems like they have two kinds. So they originally they have the naive status, and then once they're activated, it may be better to stay only activated one, not the whole population of macrophages to get better selectivity. So we found one other probe like that. 16th compound we developed, and with the gray color, CD16 binds to nicely to the active the macrophage site, so-called bone marrow atherosclerosis site, which is a vulnerable position. Now after that, we realized that there are activated macrophages of two kinds, M1 and M2. M1 is aggressive attacking policeman. M2 is the strangely changed macrophages near to the tumor. So this is aggressively attacking. This one actually suppressing the immune system, helping the tumors. So, but in this case, CD16, it binds to both of the M1 and M2. Looks like the phenotype is opposite, still activation pathway shares something in common. So our next target was we wanted to get M1, M2 selected ones, and monocytes circulating in the bloodstream. Once they have a problem, they enter into the tissue. It's before they are activated, they are coded in zero. And then they can be developed into aggressive M1 and passive M2. And when we studied those, and we realized that M1 more preferably take up carbohydrates as a nutrient source. As a nutrient source, we have amino acids, carbohydrate, and lipid. So amino acids may be common, and among those, the two other options, M1 prefers a carbohydrate, M2 prefers the lipid molecules. And then it seems like carbohydrates are taken through group transporter, and then lipid are transported by CD36 other transporters. So for targeting those specific part, we tried to make carbohydrate-based fluorescence molecules. Carbohydrate plus fluorescence motif, and then made a small library. And then when we tried, fortunately we found one dye bind only to M1, not M2, not M0. Compound structures like this. Glucose backbone, and then Cy5 structure. Now funny thing is, glucose is this size, Cy5 is almost a double size. Still, seems like a group recognizes this part, and then take up the whole thing in. So to prove that whether, and this compound we call CD17 with the red color, CD17. And first thing, to prove whether they are really uptaken by group. And when we analyze by adding excess amount of glucose, if that is really goes through the glucose transporter, it should compete with the natural glucose. If you add more glucose, it seems like they're competing out. Here, one important control experiment is glucose is important nutrient for our body. At the same time, high glucose is always a problem. Diabetics is induced by extra high glucose. Glucose is a toxic nutrient, which has an aldehyde. So as a control, we tried to add enantiomer of this D-glucose, L-glucose, chemically identical. So if they have a chemical toxicity or chemical problem, then L-glucose should not compete. And we found that L-glucose does not compete at all. So that means this is not chemical shock. This is a biological event which can be recognized for D form of glucose. And next trial is by adding inhibitor for the glucose transporter. It's reduced. Seems like it's okay, but after that we found that there are not only single group. Glucose is very important for our body. At least our body has 13 different kinds of groups. But fortunately, seems like when we analyze the M1 and M2, M1 has a rich group much more than the other. Realize that only group 1 is highly expressed in M1, not the others. It's the others that are opposite. So then we could narrow down the target to group 1, and then group 1 selective inhibitor and also a suppressor. So this example says that even though there is a group 1 in M1, if you add the shuttle to the mouse, then this probe cannot get in. In many cases, in this chemical biology approach, to prove that the group is really your target or not, another control experiment is, this is a group gene. You knock it out using CRISPR technique, then remove this, then this mouse is removed. Then check whether the mouse number is reduced and the uptake is also reduced. You removed only single protein, then uptake is removed. So that proves that group 1 is really your target. So this is one example of the group, the M1 probe, and then demonstrate that it can stain this rheumatism on the site of the mouse. And after we found the M1 probe, why not M2? And we also have the clue, M2 take what? M1 take the carbohydrates, M2 takes the lipids. Okay, then we tried to make lipid-like molecules with a fluorescence motif in the center. So in this approach, we tried to put fluorescence molecule in the middle and different size of a head group and different size of a tail group. And using this and set up the screening, M2 over M0, M2 over M1, and then try to compare those selectivity. Red color means M2 selective. Blue color means it's out. And among them, we found that one compound standing out, which stains M2 selectively, and structures like this. If you look at this part, this looks like lipid molecule, fatty acid, but using this, and we dissect the biological system, recognize this as a lipid molecule. And after we add this, and then this molecule name is a CD green 18. 18 is a compound with a green color. And then it shows that M0 not stained, M1 not stained, M2 is a selectively stained like this. And then by doing those experiments, and which transporter is important or not, and we figured out SH27A3 is a major of the player to take up the lipid molecules. And this protein is selectively expressed only in M2. Using this amount, M2 take up this compound. And if you mix up these two, green 18, green, red 17, red, and if you mix and then add to the cells, M0 does not take up any of them. M1 take up only red compound. M2 take up only green compound. So that means if you have a mixtures of M1 and M2, now it's a chance, you have a chance that you add this cocktail. Then you can count how many M2 cells and how many M1 cells there. Right? So and then another possibility, we learned 3 kinds of anti-cancer approach. First one, chemotherapy. What is the second one? What is the second generation of cancer therapy? Specific target, signaling targeting molecule. Third one, immunotherapy. We have learned. Now, people say the fourth generation, so another kind of immune regulation. Now, this one is the M2, this one is the M1. This is the old policeman. This is seems like they are on the side of the tumor right now. If you change the fate of M2 into M1, they are already near to the cancer. And we just persuade them, okay, come back to our side and attack the tumor. If you change M2 into M1, then it can be new strategy to cure tumors. So that's kind of a fourth generation of therapy, people say. There are some preliminary drugs that are developed. So we thought maybe we can monitor those changes, M2 into M1. So for this, we added that kind of a known drug, which is known to convert M2 into M1. And then we added this probe together. So here, if you don't add this drug, then most of those cells are the M2. If you add this drug, now they are changing their fate into M1. So then you can monitor those changes. And then here, one thing that you can notice is whether those cells are, okay, today I was a spy, and I'm one step moving to policeman, and then slowly changing, then they should change those colors from green to yellow and red, right? But if you look at those data, what happens? It's not the case. Seems like those changes are abrupt. And some says, okay, I changed my mind, I'll go back to the M1. And then maybe they persuade the nearby other friends, and then chunk by chunk they're changing. That means those changes are, it's a slowly, not slowly changing like this, it is changing like this. They decide whether to move back or not, and go to the M1. Okay, this is the examples I showed you. So with the time limit, I didn't show all the possibilities, but I showed you 2 examples. One possibility, maybe there is a binding target. It's kind of a conventional idea, so-called the magic bullet. If it's a drug molecule, you need to have a binding target, either receptor or enzymes. You need to activate or inhibit those targets. But in probe discovery, according to our experience, seems like this binding case is about 20% overall. And we had a very hard time to figure out the mechanism. And then later we found that this gating mechanism, either pump out or pump in. If those transporters are differently expressed, then we can discriminate the different type of cells. Later we found even those membrane structure, membrane is flexible or membrane is rigid, depending on those difference, some cells take out much better the probes than the other. In this case, this is lipid-oriented method. Another, the D class is a mechanism. Compound is taken into the cell. Only specific enzymes change that non-fluorescent one into fluorescent ones. So that's why this target cells are on their own. So at least so far we found 4 different mechanisms, and it looks like these guys, gating and the mechanism is occupying maybe 67% of all the cases. And this is about 20%, this is very minor proportions. So using this, the new mechanisms, now we are trying to attack aging. So there are the aging cells aging, so we believe this kind of our fate. We cannot avoid aging and dying. But recently, more and more, the people start to believe that aging is like a disease. Even WHO, they gave the disease code to the aging. Once you define aging as a disease, that implies that we can cure it. If it's a cure, not only it's a slow down, and even you can cure it, you can revert, and you can make the body even younger. It's a reverse aging, maybe possible. So using this approach, at least there are the young cells and older cells we can prepare, maybe easy. And then older cells can be selectively stained. And you may have heard those the laser the therapy to make your skin younger. You go to the hospital, and then under the skin, the long wavelength IR the laser killed those cells under the skin. And those empty space is filled with the stem cells. But in this therapy, you kill the most of those cells. Then even the stem cells can be killed too. So you cannot repeat this therapy over and over, maybe 3-4 times, then you cannot do any more. So now, if you have a chance to stain only older cells, then you put those cream in, then only older cells are stained, and you get to the sun tanning, and only older cells can kill. It's a selectively remove those the older cells. Is kind of a popular way currently if you have the tool to selectively kill older cells. That's ongoing right now. So this is some example of the cell selective for development and then its applications and mechanisms. Question. Okay, next week. So 어쩌다 보니까 today is the last class. That's why you had the pizza, and seems like you look very full and sleepy. And next week is our final exam. Seems like Jenny has a problem with the exam time. So what's your problem?
Actually, I don't have a problem.
Oh, you don't have a problem.
No, because I thought that the other class exam was supposed to be in 2, and it was, but it was changed already.
Okay, good.
4.
Okay, so then next, not Tuesday, Thursday. Tuesday is the Kenchunyo, it's a holiday. So next Thursday, 2 p.m. Everybody is okay for the class, for the exam? Okay, 그러면 다음 주 목요일 오후 2시에 the exam will not cover everything. The exam will cover after midterm exam. Second part we have learned. So half of the material we have learned during the second part of the class. Okay, that may be easier for you. And your friend's presentation will be included in there. So I will upload everybody's the presentation file. Now again, if you have any expected, presumed, the final exam problem set, if I like it, then I will put it up. If I put it up, then means I like it. Then it's better you look up. But you need to give your friends enough time to look it up. So if you have any the summary of those class or expected problem, send me by Sunday night. Okay? By early Monday morning, I will put it up to allow everybody has a chance and time to look up to those problem sets. Okay? Any other question? Okay, 수업은 끝내고 그러면